Hello, let's transcribe some eukaryotes. So that's you, you're a eukaryote. So we're going to talk about eukaryotic transcription. Remember, we're just making a copy. This is just the photocopy machine, so we can take the copy out of the nucleus. We're going to talk about some RNA polymerases, and we're going to contrast the three type of RNA polymerases, and they are named 1, 2, 3. Yeah, they are. Polymerase 1, polymerase 2, polymerase 3. Good names. Love it. And we're going to talk about transcription factors. Okay, let's get into it, shall we? The most important difference between prokaryotic and eukaryotic transcription, or copy making, if you will, is the copy, the photocopy that we're making has to be prepared to leave the nucleus. This is not an interoffice memo, right? We got to package that thing. So keep in mind that a lot of this, the things that we're going to do are to make sure that our transcription is going to be packaged. So we're actually going to talk about the processing in the next chapter, but just know that some things have to happen to allow it to leave the nucleus. The other thing that has to happen is, if you remember, with our bacteria, they're just all in there willy-nilly. There's a bunch of processes going on at the same time. It's all fine. It's just like chaos, and then mRNA comes out, right? Eukaryotic is a little bit different because it's wound around a histone, right? If you remember. Okay, so if you can imagine it, your DNA, here's your amino acids, here's your whole code. DNA is wrapped around these histones, okay? So it's wrapped around actually 3, 4, right? It's wrapped around like this until it makes a chromosome, right? I know that looks like a mess, but that's how it looks. So we can't transcribe this until we unwind it. Because do you see how that sometimes it overlaps and you can't get in there? You can't get into this, so you have to unwind it. So that is one of the biggest differences, is that you have to unwind your eukaryotic DNA and you have to process it before it can leave the nucleus. Okay, so you have to transport it into the cytoplasm and you have to protect it from being degraded and you have to unwind it. Okay, so let's get into it. 3 different polymerases named 1, 2, 3. I feel like you should be able to get this right. Name the 3 polymerases. 1, 2, 3. You can use Roman numerals. Okay. Eukaryotic mRNA are typically monogenic, meaning they specify for a single protein. Now, I want to explain to you this because earlier we used the vocabulary word monocistronic. Monocistronic means a single mRNA codes for multiple proteins. Monogenic means one disease, one trait. So monogenic is like when we talk about peas, those are monogenic. If you have this gene, you're a yellow pea or a wrinkled pea or a smooth pea. That's monogenic. You have it, you don't have it. Polygenic is like skin color, is diseases that have a spectrum, right? We don't have just 3 skin colors. We have a variety of skin colors. So those vocabulary words get confusing because monocistronic and monogenic, both monos, one means monogenic. If you have the genotype, it only codes for one phenotype. So if you have the gene, you get the disease, or you get the color, or you get the wrinkled seed. Monocistronic means it is this where we're talking about each mRNA only codes for one very specific protein. Okay, I just wanted to take it aside and say, hey, these are 2 different vocabulary words. So be careful. Okay, back to our story. We are transcribing eukaryotic DNA into mRNA. So the first thing that we need, again, is initiation factors. In prokaryotes, they are called transcription factors, and it is a whole bunch of things. Look at all this magic happening. But it's also, don't panic, fairly similar to prokaryotes. We got a little bit more going on. You've got your RNA polymerase here. You've got a promoter, and you've got what are called transcription factors. There's a bunch of them. I'm not going to make you memorize them. You don't need to know them yet. You can save that for a more advanced class, and then you can dive deep into all the transcription factors. You are also going to have enhancers and activators. But let's talk about the good old polymerases, okay? There are 3 polymerases. They're each made up of about 10 subunits, and each eukaryotic polymerase also requires a set of transcription factors that bring it to the DNA template, right? So not only do you need the RNA polymerase, but you need a transcription factor that brings it, right? You need something that brings it together, okay? So let's talk about RNA polymerase 1. We start with 1 because that feels like a reasonable place to start. This is located in the nucleolus. It's a special nuclear structure, and it's associated with rRNA. I'm not stuttering. R, little r, big RNA. That's ribosomal RNA. That's this big hamburger thing that's going to squish our mRNA into a protein, right? It's going to bring these tRNAs, which we'll talk about. So your mRNA, your rRNA, here's your ribosome. rRNA is, it kind of looks like a cheeseburger to me. It's this big structure that's going to bring your tRNA together with your mRNA. This is your mRNA. This is your tRNA. And this big, it looks like a skull, hamburger, whatever you want to call it, that's your ribosome. Made out of rRNA. And you need polymerase 1 to make that hamburger magic happen. Okay? Okay, so again, before we move to the next slide, polymerase 1 is located in the nucleolus, and it's associated with rRNA, which is the ribosome. All right, polymerase 2, located in the nucleus, and this is going to synthesize our pre-mRNAs. So this is what is going to synthesize our mRNA into the RNA that is going to leave the nucleus. So for that, we are going to need RNA polymerase 2. They undergo extensive processing after transcription, but before translation. And RNA2 is responsible for transcribing the overwhelming majority of eukaryotic genes. So RNA2, very important, and is associated with your mRNA. Guess what's coming in at number 3? RNA polymerase 3. This is going to be associated with our only friend that is left, tRNA. Okay? This is the transfer RNA, and it looks like a T, and we will talk about it. The tRNA's role is to bring your amino acid and code it with your 3 codons. It's going to read the codon, and it's going to bring the amino acid that is necessary. Okay? Small nuclear RNAs, they have a variety of functions. Another thing that polymerase 3 does is it splices the mRNA and regulates all of the transcription factors. But we will get to that. Just know that polymerase 3 is involved in that. And here's a handy dandy chart, right? Polymerase 1, nucleolus. rRNAs, polymerase number 2, mRNA. Polymerase number 3, typically tRNA, okay? Now, this is just information for you. So I'm not going to ask you this down here, 1, 2, or 3. I'm just going to explain to you what they're talking about. So when you hear these words, you know what I'm talking about. And because this looks like toadstool from Mario, right? The little toadstools, this is what they're talking about. So if you are characterizing a new gene, which we will be doing potentially in lab, we will need to determine which polymerase transcribes it. We will test this with the process of alpha amyloidin. All right. I'm having trouble saying it today. Anyway, it's a toxin that's produced from this toadstool. Okay. This toxin is insensitive to 1, moderately sensitive to 2, and extremely sensitive to 3. So when you expose it to this toxin, it'll stop whichever one of these things it is the most sensitive to. And this is how we can tell which polymerase it is. Okay, I just think that's a fun fact, and this looks like toadstool. And this is what we're talking about when we're talking about polymerase sensitivity, exposing it to the mushroom. Okay. Polymerase 2 promoters and transcription factors. Okay. Similar. Again, we've got a Tada box, we've got a promoter, and then we've got these little transcription factors. See how they're TF, transcription factor. See all these little transcription factors? And once you get all of the transcription factors, what are you going to get? Your RNA polymerase 2. Because your RNA polymerase 2 is responsible for your mRNA. This is your mRNA, okay? Again, this is about 10 sequences up, and there is a Tada box involved. And then here is where our transcription start site will be. The polymerase 2 and the promoters are going to be further up, and they are located also 25 to 35 bases relative to the initiation site. And the thermostability of ANT bonds is low, so it doesn't take as high of a temperature to break those AT bonds, A to T. It doesn't take as much, so the thermostability of those is low. And this helps the DNA template to locally unwind in preparation for transcription. So that's what the Tada box is for, because it's a TATA box, and it's going to denature or unwind quicker. Okay, and here's a little bit of that story. Here's your Tada box. Here's your transcription factors. All of this is about 30 nucleotides upstream, and then you have your transcription factors and your RNA2. Okay, instead of the simple factors like the sigma factors that we had in eukaryotes, we have transcription factors. There's a lot more of them. They're a lot more complicated. They are, again, required to recruit polymerase 2. Why polymerase 2? Because that's your mRNA. Polymerase 1 and polymerase 3, we're going to talk about when we get to translation. But primarily in transcription, we're talking about polymerase 2. Transcription factors are also called basal transcription factors when they finally bind to the promoter. Just another vocabulary point of fact. Some eukaryotic promoters also have a CAT box, GGCCAATCT, and that is approximately 80 nucleotides down. Further upstream from that is going to be a GC-rich box or an octamer box. And you're just going to see these things. I'm not going to ask you to find them unless they're already there. The GC box, the CAT box, the Tada box. Okay, you can see that they're there. And these are regulatory promoters, because we, as a eukaryote, we don't want just things transcribing willy-nilly. We want this tightly regulated. So we have all of these promoter components before we even start transcription. Okay. Again, transcription factors. There are a bunch of them. So the complexity of this transcription does not end with just the polymerases and the promoters. There's an army of transcription factors. You do not have to memorize them. We're not doing that. I just want you to know that they are a bunch of transcription factors. There are silencers. There are enhancers. They increase the efficiency. They slow down the efficiency. Some of those enhancers and silencers just affect how fast or slow it goes or the efficiency of it, but some of them are not necessary. So you have activators or enhancers, and this affects the speed of transcription. You have repressors. These bind to the selected sets of genes and they silence or slow transcription. The basal transcription factors, remember, in response to injunctions from the activators, they position RNA polymerase at the start. So once you have everything in place, you have basal transcription factors and co-activators. These adapt to signals from activators. So there is just so many things going on with the transcription factors. I don't want you to be bogged down with the names of the transcription factors. Just know there's a bunch of transcription factors. They slow things down, they speed things up, they stop things, they start things. All of these are transcription factors, okay? A lot going on. Okay. Bringing RNA polymerase 1 and 3 to DNA involves simpler promoter elements, but that's not going to happen until we get to translation. I've not forgotten about polymerase 1 and 3, but just know you see them when we do replication, right? 1 and 3. Right now, we're not doing that. We're just doing transcription. We have not gotten to translation yet. So we don't need the polymerase 1 and 3 quite yet. Again, it's a tightly regulated process. Okay, following the formation, we have our initiation factors. Everything is happening. What way does it go? Come on, you know this. 5' to 3'. We need to start on that 5' carbon, that carbon that's sticking out, okay? That's the 5' carbon, so it's synthesized 5' to 3'. We don't need the Okazaki fragments because we only need one copy of mRNA. We don't need two copies. We're not copying both like double-stranded DNA. RNA is single-stranded. So you only need one copy. So that's why we're not making Okazaki fragments. Yes? Okay. Elongation is the crucial stage where RNA polymerase moves along the DNA strand from 3' to 5', and it makes the complementary strand 5' to 3'. Just pause it, think about that, right? It moves 3' to 5', it makes it 5' to 3', okay? Moves one way, makes it the other way. Again, A is going to go with U, and it's going to build a new growing molecule 5' to 3', forming your copy or your RNA transcript. Okay, now what's the last thing I said we need to address? We need to address this winding, right? We need to address all of this on the histone, right? This is the histone and it's wound around there. So although elongation is essentially the same in prokaryotes and eukaryotes, the biggest difference is you have to unwind this DNA from your histones. Okay, you've got the DNA, they are wound around histones. That is wound again until you get chromosomes. So elongation can't happen until that histone complex is dealt with and you have an unwound portion of DNA. It is called the DNA histone complex. See how we're naming these things? They make sense? So just remember, okay, you've got histone modifications. Right? In order to get this away from here, you've got to unwind it and wind it. Okay? So, some of the ways you're going to do that. Methylation and acetylation, right? Methylation. Basically, acetylation is going to add an acetyl group. Okay? The acetyl group to histones removes their positive charge. Right? If this is positively charged to my finger, it's going to remove that positively charged and it's going to weaken the attraction so it can unwind. Deacetylation removes the acetyl group's attraction and condenses them sometimes into silent genes. Okay? It increases, right? It winds it around more. So acetylation loosens it. Deacetylation winds it closer. So basically it can silence the gene because now all my genes are on my chromosome. How are you going to get there? Acetylation gets it there. Okay, some other things we have are called FACT, which are facilitates chromosome transcription. Don't you love these names? They actually just mean what they do. FACT temporarily removes histones for access to modifications, sort of like acetylation, and they loosen their grip. Okay, so you have acetylation, you have methylation, you have deacetylation, and you have some of these are facilitates chromosome transcription. So they're FACT factors. I don't love that. I don't love that word, FACT factors, but just know that FACT is facilitates chromosome transcription. You also have chaperones that will restore that nucleus structure after. Okay, so once we have our acetyl group, our histone, and we've unwound this, the FACT complex replaces the histones and it recreates the nucleosomes. The termination of transcription is different from different polymerases, right? Generally, it involves the release of the transcript and the polymerase from the DNA. Generally, we're kicking off the DNA polymerase, just like we did with eukaryotic genes. So the gene is transcribed transcribed into RNA polymerase 1, and it recognizes a termination protein, and that termination protein, again, is like a hairpin. Do you remember when we did the hairpin? You can go back and look at that video when we talked about prokaryotic termination, and one of those was a hairpin, in which it involves the termination because it kind of just kicks it off. That hairpin, right, makes a loop, and then it knocks it off, okay? That's exactly what happened. The terminator, hairpin formation, and transcription knocks it together. Okay, there is the summary of it. We looked at transcription, the 3 types of polymerases. Really, we concentrated on polymerase 2 because that's what's involved in transcription. We talked about transcription factors. We talked about promoters. And we talked about removing the histone so that we could get to the DNA and transcribe it. And that is the basis for eugenics, really. That's what we're doing with that eugenic. Now, I want you to stop the video here and make sure you can list the steps in eukaryotic transcription. You can discuss the role of all 3 polymerases. You can compare and contrast the polymerases. You can explain the significance of transcription factors and histones and facts. Okay.